Hi everybody, we are going to go through section 1.4.2 to understand how we do the recombinant DNA part. So really in your gold packet, you should have gone through part 2 where you marched through the interactive to learn about recombinant DNA. So it had you go to the revolution part, the problem, and understanding the techniques about how scientists can actually make recombinant DNA. This is done all the time for various purposes to take a piece of DNA that we actually care about and put it onto a plasmid, if we remember what those are, those small circular pieces of DNA. And then we can use them for different purposes. We can put them into different organisms. We can use them to make vaccines in this case, and we'll have lots of other applications that we'll learn about later. But the idea is there is cutting and pasting involved. And then this picture, if I can kind of zoom in, shows you a little bit about what that looks like. But I'm going to march through how you're going to actually make your own in this class. So to do that, we're basically going to go to the identify restriction sites, and I'm going to march you through the rest of the activity. You're going to need this sheet of paper. So at the top, you will see there is a chart. And in that chart, there are a number of restriction enzymes. So restriction enzymes are basically tiny little enzymes that are scissors. They're the molecular scissors that you learned about before. Their job is to go in and cut DNA at various points. Next to the name, so there's hundreds, thousands of different enzymes that exist. These are their names. Usually it has to do with a particular bacteria or virus that they come from. So there's one called BAM-HI, ECO-R1, HEY3, HIND3, and PST1. Those are the ones that we're going to be using today. And then next to it, it says the specific sequence that they're looking for. So usually there's a 6-letter DNA sequence. 6 letters is usually long enough for the enzyme to be specific to a piece of DNA. So it's just scanning all of the DNA to find that particular sequence. And when it does, it's going to cut that sequence in a particular pattern. There's 2 ways to cut. There are what we call sticky ends, which cut in a non-straight line. So you can kind of see here on the first one, it's cutting, if I zoom in a little, it's cutting after the G, and then it zigzags over and cuts next to the G on the other side. So when it cuts in this zigzag pattern, it basically creates these little overhangs, or sticky ends as they're called. And that way when you're trying to glue the DNA back together later, it's a little bit easier because you have these little overhangs and then they can kind of grab onto each other. And you can see they are complementary. So when you're trying to glue these things together, they're complementary, so they'll want to stick together. The other option is what we call a blunt cut, and that's like this one right here. It finds the sequence GGCC in this case, and it just chops right down the middle. That blunt cut works. It's just a little bit harder to glue together. It's like if you had 2 surfaces or pieces of wood, for example, and you just try to glue them together like this. It's a lot harder than if they had overhangs and more surface to really stick together. So those are the enzymes, and those are the sequences they cut and how they cut. So that's important. At the bottom of the sheet, you're going to see plasmid strips. So the first 2 rows are one continuous strip. So what we're going to end up doing is cutting this thing out. You're going to glue the, or tape, I would say, the second row right onto the end of the first row. It's just one continuous strip. And then we're going to circle it around and make a plasmid, because plasmids are small circular pieces of DNA. The bottom contains the viral DNA. So when we're making a vaccine, in this case, we're going to take a small piece of viral DNA and insert it into the plasmid. And we're doing this so that when we use this piece in a vaccine, the small piece of viral DNA is enough to trigger our immune system to actually have a response. And that will be what kind of gives us our immunity to that particular disease. And it's not enough to give you the full-blown disease because it's only a little small chunk of DNA. So how do we go about doing this? Well, the first thing that we need to do is identify the restriction sites. So in your sheet, what I would like you to do is basically assign a color to each one of these sites. So that's the first step, assigning a color. I'm just going to go through, for example, and say that BAM-HI is going to be purple. And I can show you kind of how it cuts like this. Great. And then I would pick a different color for each one so that I end up with something that maybe looks like this. So I have different colors for each one. I'm showing you roughly how they cut. You can do it in the middle column or the end column, doesn't really matter. But that's really what I'm going to be looking for. So now I'm going to use that information to scan through the DNA sequences. I'm going to go through all 3 strips and try to identify those parts. So for BAM-HI, I'm going to look for GG-ATCC. And looking along the way, I see one fairly close. I'm going to highlight all of the ones that I see right now. And you're going to want to highlight both the top and the bottom strand. So there's one right away up top, the GG-ATCC. And then the one on the bottom that goes along with it too. Those are the complementary strands. And if I'm looking at how it is going to be cut, I can kind of draw that in as well. So you can kind of see how I did that. Now, that's it for the plasmid. There's no other pieces in the plasmid where that sequence comes up. Now, down in the viral part, you're going to see that it shows up a couple of times. So you're going to want to just color code and work your way through. You're going to want to do this for all of the different restriction enzymes that could possibly go through. So this is a little bit more advanced version, but I did all the different colors, how they cut, and then at the bottom you can kind of see these are roughly where your sequences are going to be. So there's different colored ones throughout. So that piece of it is important because you're going to be literally cutting and pasting this thing together. So that's how you go through and identify restriction sites, which brings us to now choosing the right enzyme. Or what I would do intermediate to that is I would take this piece and I would cut it out. So once you have it all color coded and labeled, go ahead and cut your strips. So you're going to have 3 total strips. You're going to have a viral DNA strip. Probably keep the label of viral DNA at the top so that you don't accidentally get it confused. And then you're going to have a plasmid strip A and a plasmid strip B. As I mentioned before, the 2 plasmid strips are really going to be end to end. So A comes first and then B. If it helps, you can kind of trim off the end of plasmid B. Grab the tape here. We're going to tape it together. So I'm just taping A onto the end of B, like this. So it's really one long continuous sequence. But circling it around, it's actually a circle because it's a plasmid. So I just kind of trimmed the end of that one again. So the sequences should line up nicely. Voila, I have a plasmid. And it should be fully labeled with all of the different restriction enzymes that you have available to you. All right, so you have your plasmid and it's been labeled and it's now a circle. The key part here is to really figure out where you can cut this plasmid to then be able to insert this viral piece of DNA. And you can't just chop this thing anywhere. There are important pieces on here that are needed in order to have the plasmid successfully copy the DNA and make the protein or whatever it is that it needs to make. So there are key features. And those key features are listed in here under choosing the right enzyme. So our goal is to pick the one. In our chart, we have 5 different options that we can choose from, and we know where it cuts. So we're going to use the properties of the plasmid to try to figure out which one is the best one and why. So we have all the sites labeled. We're going to pick the one. It says in step 18, scan the plasmid DNA as well as the gene from the hepatitis B virus, because we're making it for Hep B, find the appropriate enzyme. The first bullet point that's really important. Use an appropriate restriction enzyme to open the plasmid ring, as well as cut a piece of viral gene. Do not use an enzyme that digests it into many tiny pieces. Why is this important? If you chop a plasmid up into lots of tiny pieces, it's really hard to glue it back together in that correct order. It can get all mixed up, and if it does get mixed up, then it can't do its job. So you want to scan your plasmid ring and identify one that does not chop it into a lot of pieces. So looking at your colors, see if you can pick out which one is the right one to not use. Can you look at this and eliminate one that chops it into many different pieces? And the answer is yes. Yes, you can. So see if you can figure out now which one that is. All right, so once you've eliminated that one, put a little X next to it on your table, because that's not going to be one that we want to use in this case. So I'm going to do that for the one that I know that it is in our particular scenario here. All right. So the next bullet point. Do not cleave means cut. Do not cleave more than 20 base pairs off of either side of the viral DNA. So when you're looking at your little piece of virus DNA that I have color coded here also, you don't really want to cut more than about 20 base pairs off either side. Because the idea is this is a gene. You do want it to express. And if I chop it in the middle, then it's not going to be made in the functional form that it needs to be. So if you chop a little bit off the ends, usually that's okay because the gene is in the middle. But if you're chopping in the middle, that's a bad sign. So look here and see which ones of your color coding system would you now want to eliminate, and see if you can put an X next to them right now. So I am doing that now. I was able to eliminate 2 different ones, which brings us to our next bullet point. Choose an enzyme that does not interrupt the antibiotic resistance gene in the plasmid or the origin of replication. So those are 2 parts on a plasmid that usually come standard. So in our little plasmid strip that we have, looking at it, there is an antibiotic resistance gene, as well as if you look to see somewhere above the top, there's something called the origin of replication. So the antibiotic resistance gene is something that we've kind of learned before when we talked about conjugation of plasmids with antibacterial resistance. Scientists use that to select for the successful ones. If the bacteria that we're trying to grow this vaccine up in is resistant to whatever antibiotic is on this plasmid, when it isn't normally, then it will grow in that situation, and we know that those are the ones that have worked. So the gene itself extends across the top wherever you see that dotted arrow. So if there are any enzymes that cut in there, that's bad news. So looking at my little sheet here, I can easily eliminate one as an option because it cuts in the middle of that gene. So go ahead and eliminate that one if that's one that you had to label. Also, you don't want to cut in the middle of the origin of replication. So that's right here for me. And you can see there is one that does cut in the origin of replication. So that's a deal breaker. Double check that you have crossed that one off of your list. Let's see. And with that, the last bullet point, make sure you create sticky ends. So sticky ends, as I mentioned, are really preferable. You want it to overlay so that we can glue this thing back together. And so looking at all of my different enzyme options, looking at the ones that are blunt cuts, so the ones that just cut a straight line through, are not going to be ones that we want to use. So if you haven't crossed that one off your list, then you should. So that concludes the bullet points. And now on your list of enzyme options, you should be able to identify the one that's going to be the one that we want to use because it really fits all of the different criteria. Great. Now we actually do that. So you have your plasmid strip. You are going to identify the restriction site. So which one of these guys was the right one? You're going to find it, and you're going to actually cut the strip according to how it would cut. So if it creates a little jagged pattern, I'm just going to arbitrarily pick a spot for this right now. Let's say it's right here and it makes a little jagged cut. I'm going to take my scissors and I'm going to say, okay, G, and then I'm going to zigzag it up and I'm going to make my little cut like this. So let's say that's where the spot is. You can see that I've kind of created these sticky ends that can then overhang. You're going to take your viral DNA and you're going to do the same thing. There should be sites on here that for the enzyme that we're going to use. So you're going to take that same principle, you're going to take it and you're going to cut wherever that may be. So going through, cutting, cutting, cutting. If you have little overhangy pieces, that's okay. It's just garbage, essentially, at that point. But you've cut your plasmid. Now it essentially can open up, and you can take your viral piece of DNA, and it should now be able to overhang. You see the sticky ends, and you're going to tape that guy right in. So essentially, we've created a slightly larger plasmid that now contains our viral piece of DNA. And that's essentially what recombinant DNA is. So here's kind of my final one. You can see that I had all the restriction enzyme sites labeled. It's not cutting into my origin of replication. It's not cutting into my antibacterial resistance gene. And you can see I've colored the viral piece of DNA kind of in purple, so you can see where it ends up going. So this is my recombinant DNA vaccine that now I could be used to put into a person, and then this piece of virus would be made, our body would trigger an immune response against it, and then we would be immune to that disease. Okay, I hope this video helped you put it all together on your own, but we'll see. Thanks.